Hi, my name is Antonia and I'm a PhD student at Aston University working on mycobacteria drug discovery. My project aims to create a new in vitro model which replicates the physiological conditions of the non-replicating persistent form of Mycobacterium tuberculosis using droplet microfluidics. Tuberculosis is a disease of the lungs caused by Mycobacterium tuberculosis, and it can survive in a dormancy state known as non-replicating persistence. The external pressures such as hypoxia, nutrient deprivation, low pH and a high CO2 created by the host environment induces a survival response of the bacteria and an encapsulated granuloma. This physically restricts penetration of antimicrobials and thus provides protection. The NRP state can also relapse into active disease, especially in high-risk groups, such as immunodeficient individuals. Not only do we need effective new antimicrobials, but we also need to advance the drug discovery pipeline by developing new innovative drug susceptibility assays which replicate the biological host. To do this, engineering approaches such as applied technology can advance the field of drug discovery. Microfluidics is a rapidly growing field which comprises multidisciplinary expertise which uses the systematic manipulation of systems that have microscale channels where fluid volumes of nanoliters to attoliters can flow in geometric configurations. Well suited to the scale of bacteria, microfluidics can produce biological assays in parallel with well-defined controllable environmental conditions. Droplet fluidics is a subset of microfluidics in which nano to femoliter volume droplets can be routinely generated by droplet making micronozzles in a carrier fluid at production rates exceeding 10 kHz. It uses an aqueous and oil phase to generate droplets. I will be using pico-droplet single-cell encapsulation system device, which can accurately control pico-droplet production through an imaging-based feedback and pressure-driven flow. This will maintain monodispersity at the high pico-droplet production rate and enable the encapsulation of bacteria and antibiotics at high throughput, which replicates the NRP environment through hypoxia and nutrient deprivation by using our anaerobic cabinet and minimal culture media. The droplets will be generated on PDMS chips using standard soft lithography techniques with an oil inlet, bacterial culture inlet and an antibiotic inlet. Using the flow focusing geometry, droplets will be created which has encapsulated bacteria and antibiotic. The droplets will be collected from the outlet for off-chip incubation and subsequent imaging analysis for bacterial proliferation. This provides possibility of undertaking millions of experiments within a single microfluidic device, providing high throughput assay for antibiotic drug discovery. The gas pressure system and picoliter scale of droplets also supports the use of much smaller volumes of sample than standard susceptibility testing, and thus it reduces their reagent amounts and preserves samples. The compartmentalisation of single cells from high density cultures also could prevent competition for nutrients or space among mycobacteria, allowing the slow growing species to proliferate and narrowing down the assay time. I'd like to thank my supervisor, Dr. Jonathan Cox, my industry partner, Sphere Fluidics in my lab, and all the students and mentors from the CDT.